Hello, and welcome to Progress in DM2 Research from the Disney Lab in RAN proteins as a therapeutic target. I'm Clede Cumming, MDF's Communications and Technology Manager. This session is part of the DM Professionals track, which is intended for clinicians, researchers, and industry experts. This session will be available for viewing after the conference at the MDF Digital Academy at myotonic.org/digital-academy. I'm delighted to introduce our two presenters. Our first presenter is Dr. Monica Banez Coronel, an assistant scientist at the Center for Neurogenetics at the University of Florida. She completed her PhD in biochemistry and molecular biology at the Autonomous University of Madrid and held a postdoctoral fellowship at the Center for Genomic Regulation in Barcelona, where she explored small RNA-mediated toxicity in Huntington's disease and the suitability of blocking RNA toxic species to improve disease symptoms in mouse models. Shortly after the discovery of repeat-associated non-ATG or RAN translation by the Random Lab at the Center for Neurogenetics at the University of Florida, Monica joined their team and focused on studying RAN translation in Huntington's disease, RAN protein toxicity, and the correlation of RAN protein accumulation with disease pathology. Monica joined the faculty of the Department of Molecular Genetics and Microbiology at the University of Florida in 2017. Her current research, which she will discuss today, focuses on exploring RAN translation accumulation across CAG-CTG expansion disorders and understanding the contribution of RAN proteins to disease pathogenesis. Our second presenter, Dr. Matthew Disney, is a native of Baltimore, Maryland. He received his early schooling in the Baltimore Catholic school system, his Bachelor of Science from the University of Maryland, and his PhD from the University of Rochester in physical chemistry. As a graduate student under the guidance of Professor Douglas H. Turner, he studied RNA catalysis and developed approaches to determine RNA secondary structure in cells using a combination of experiment and prediction. Matt completed postdoctoral training at the Massachusetts Institute of Technology and the Swiss Federal Institute of Technology in Professor Peter H. Seeberger's lab and helped develop the glycan microarray technology. Matt is currently a professor in the Department of Chemistry at Scripps Research. His laboratory has pioneered the area of small molecule targeting of RNA. A central feature of the lab has been to address fundamental questions how molecular recognition occurs between RNA folds and small molecules, and to apply this knowledge to address questions of biomedical importance. Applications have included development of sequence-based design of small molecules, on-site drug synthesis in diseased cells, understanding the biology of coding and non-coding RNAs, and work on interfacing RNAs with quality control machinery that has included monomeric degraders and chimeric degrading compounds. Dr. Disney will discuss progress in DM2 research from the lab. Enjoy the presentations and welcome, Monica.
Hello, everybody. I would like to first thank the organizers for the opportunity to present our data here today in my talk entitled RAM Proteins Accumulate in DM1 Brain. So as an introduction, repeat-associated non-AUG translation or RAN translation was discovered by the Random Lab in 2011 and demonstrated that repeat expansion mutations can undergo translation in the absence of an AUG initiation codon. RAN translation occurs in the 3 reading frames. So in this example from a CAG expansion, that means that RAN polyglutamine protein will be made from the CAG frame, a RAN polyserine protein will be made from the AGC frame, and a RAN polyalanine protein will be made from the GCA frame. Additionally, RAN translation occurs from both sense and antisense transcripts. So a RAN polyleucine protein, polycystin protein, and a polyalanine protein will be made from the opposite strands. Through RAN translation, a single expansion mutation can originate up to 6 different repeat expansion proteins. RAN translation is repeat length dependent. So longer repeats normally associated with more severe forms of the disease leads to higher levels of RAN proteins. RAN translation occurs from 3 tetra, penta, or hexanucleotide repeats, and in expansion, mutations located in coding and non-coding regions, as well as 5 and 3 primes UTR. Additionally, RAN structure is important for RAN translation, and the need of RNA hairpins, stable RNA hairpins, are important for RAN translation to occur. RAN translation occurs in vivo, and RAN proteins have been reported in 11 repeat expansion disorders until now. 2 of these expansion disorders that accumulate RAN proteins are DM2, with the presence of poly-LPAC and poly-QAGR in the brain of DM2 patients, and DM1, where polyglutamine aggregates have been reported in cardiomyocytes and leukocytes from both DM1 mice and DM1 patients. So one of the main questions is are these proteins participating in disease at all? So some of the probably more clear data come from the C9 or 72 ALS FTD context, where experiments from Lia and Nguyen at the Rano lab were conducted to inhibit or target RAN-GA protein using a specific antibody. And what she could observe is that the targeting of these RAN proteins reduce the protein levels, not only GA, but also the levels of other RAN proteins, increase mice survival, improves the behavior of these mice, and also decrease or improve different markers of pathology, like neuroinflammatory markers that were decreased, and also improve the survival of spinal motor neurons. In the CAG, CTG expansion disorders, we have evidence of RAN proteins being linked to pathology. Here I show you some examples of polyserine RAN proteins levels being increased with age and disease severity in SCA mice. So there is a correlation between polyserine levels and aggregation and the phenotype of these mice. And also polyserine aggregates are found in regions showing white matter abnormalities and axonal breakage. In Huntington disease, RAN proteins are colocalizing with active caspase 3, indicative of the RAN protein positive cells undergoing cell damage of or maybe an apoptotic response. And also HD RAN proteins are found to be highly increased in severely affected cerebellum of Huntington disease juvenile cases. Based on those evidence, our main question was, do RAN proteins contribute to myotonic dystrophy type 1? So to answer that question, we developed a set of antibodies to detect RAN proteins, an antibody against the polyleucine repeat motif that will detect RAN proteins originating from the CTG expanded transcript, and an antibody against the polyserine repeat motif that will detect proteins or RAN proteins originated from the CAG antisense transcript. We chose to focus on polyleucine and polyserine first because these proteins are shown to be toxic to neuronal and glial cells in culture. And because there is ample evidence of RAN protein accumulating inside of pathology in the brain of neurodegenerative disorders caused by repeat expansion mutations. And because in DM1 itself, those patients, we know that they carry entire variety of CNN symptoms, we decide to explore the presence of these proteins in brain. But as a first step, we decided to validate or to test whether these tools, these antibodies against the polyleucine and polyserine repeat motif, were reliable and sensitive enough. And we did that in cells transfected with recombinant vectors expressing a mini gene containing an amino terminal flag tag, followed by a leucine repeat motif or a serine repeat motif. Immunofluorescent staining show that the signal from the polyleucine antibody, indicated here in green, colocalized with the signal from the flag antibody, indicated in red, while no signal were seen or observed or detected in cell transfected with the empty vector. Additionally, polyleucine expressing cells incubated with a pre-immune sera didn't give us any green signal either. The same set of experiments were conducted for the flag serine fusion protein using the polyserine repeat antibody. And again, we could see a nice colocalization between the polyserine antibody signal and the flag signal, while no signal were detected in our negative control samples. We conducted a second set of experiments to validate our antibody, in this case, using human postmortem tissue and immunohistochemistry as a technique, because that will be the sample type and conditions that we'll use later in our study. As a positive control, we use Huntington disease striatum, a brain region that we know express RAN, polyleucine, and polyserine proteins with previously as detected with previously characterized and validated antibodies that target the C-terminal region right after the repeat. So we compare the signal from this validated antibody with the signal from our novel repeat antibodies. And as we can see here in these panels, a polyleucine repeat antibody gave us the same sort of signal as the previously characterized leucine C-terminal antibody for human Huntington disease caudate. And the serine also gave us the same nuclear staining accumulation that was detected with the previous serine C-terminal antibody, while no signal whatsoever was detected in our control samples. So these experiments validate the use of our novel repeat antibodies for the detection of leucine and serine proteins in human brain, and that they are reliable, sensitive, and specific tools. So we conducted our experiments focusing in DM1 frontal cortex, a region that shows some atrophy in gray and white matter. And we conducted immunohistochemistry staining in 9 DM1 samples and in 9 different control samples. And I'm going to be showing you now the staining seen in gray matter and white matter for leucine and gray matter and white matter for polyserine. So as we can see in these panels, DM1 cortex shows large and frequent polyleucine aggregates, while no signal were detected in the control subjects. This sort of staining was the more frequent one that was mostly showing a perinuclear or cytoplasmic accumulation pattern. In contrast, in white matter, the polyleucine proteins accumulate as smaller aggregates, very frequent. You don't need to hunt for them at all, and mostly located in the neuropil. However, although these massive large aggregates in gray matter and these micro aggregates in the white matter regions are the most common form of aggregation and distribution pattern we observe in these samples, there is some variability. And here I show you some examples of regions showing smaller aggregates or soluble forms of polyleucine. White matter regions were more consistent, about always showing aggregating forms of the protein, although with more or less frequency. So what about the antisense transcript? Are antisense RAN proteins accumulating in DM1 brains? To answer that question, we conducted the same experiments using the polyserine repeat antibody. As we can see here in the panels, the gray matter regions of DM1 frontal cortex show frequent large aggregates around the cell nuclei in both neurons and glial cells, while no signal was detected in the control. In white matter regions, these aggregates were more frequent, although significantly smaller in size, and normally located in the neuropil. So looking at these patterns, it's like polyleucine and polyserine RAN proteins in the M1 could be forming part of the same macro aggregates, maybe even trapping other proteins into the same mix. So this is the more typical form of aggregation we encounter in our 9 DM1 cases: large perinuclear aggregates in gray matter regions, small frequent aggregates in the neuropil for white matter regions. However, polyserine RAN proteins was also present in different patterns, like as we can see here, with the presence of less robust aggregates, soluble forms, or even regions where it was hard to detect. In Y matter regions, the presence of polyserine was quite robust, and aggregates were frequently detected. So looking at our cases together, in the majority of our cases, we find both polyserine and polyleucine aggregating in the same distribution pattern in the same region. This suggests that these repeat proteins from the sense and antisense expanded transcript often co-aggregate, and that these transcripts are being translated at the same time. So what does it mean that these proteins are being so they're so abundantly accumulating? Is there any relationship between the presence of RAN, polyleucine, polyserine repeat proteins, and DM1 pathology? So to answer that question, we conducted another experiments using serial sections, because Y matter is show alterations at early stage of disease. We focus on Y matter regions where we stain for sense and antisense RAN proteins, and also for LASO fast blue that label myelinated axons. So as we can see here, the regions that show in DM1 cortex, those regions that show frequent polyleucine and polyserine aggregation also show some Y matter damage in the form of demyelination. Well, the control that doesn't show any RAN protein aggregates have a really robust blue dark staining. So additionally, looking at our samples, we could see an increase in the number of astrocytes in both gray matter and Y matter regions. So we also did an staining where in serial sections we label for RAM proteins and for GFAP, a marker of reactive astrocytes. And as we can see in our panels, those samples that have a more frequent and larger aggregates also show an increased number of GFAP positive cells. That was observed not only in gray matter regions, but also in gray matter regions. Indicating that those areas where the RAM proteins are accumulating in DM1 are undergoing some form of damage and maybe neuroinflammation. We confirmed that using IBA1 as a marker for macroglial cells, and as we can see here in this sample in gray matter regions, although we see the same for Y matter, those areas that show frequent and prominent RAM protein aggregation also show a high number of cells that they were positive for IBA1, not only in the ramified form, but also as the amoeboid form, more indicative of an active, maybe phagocytic process. So it seems like polyserine and polyleucine accumulation are associated with regions where there is a neuroinflammatory process. To summarize, I want to mention that novel sense and antisense polyleucine and polyserine RAM proteins are frequently found in DM1 autopsy brains. That polyserine and polyleucine proteins are toxic to cells. That these aggregates are frequently found in gray and Y matter frontal cortex regions, and they strongly associate with demyelination, providing a possible molecular explanation for the Y matter abnormalities seen in DM1 patients. Additionally, polyleucine and polyserine aggregates strongly associate with regions of neuroinflammation, including microglial activation and reactive gliosis. And future studies are aimed to understand the role of these proteins in disease and on therapeutic opportunities to reduce RAM protein expression. Finally, acknowledge everybody that has contributed to this work. The people at the Rano Lab at the University of Florida, also our collaborators at UF, as well as Johns Hopkins, University of Minnesota, and Stanford, and our funding sources. Thank you for your attention, and I'll be happy to take any question.
Hi, everyone. It's great to chat with you today. My name is Matt Disney. I'm a scientist at at Scripps Research Institute in Jupiter, Florida. I'm also founder of Expansion Therapeutics, which is has been both a clinical, but it's also a preclinical drug discovery biotech company where we're developing medicines that target genetically defined diseases, including types 1 and types 2 myotonic dystrophy. Today, I'm going to tell you about work that my academic lab has been doing on studying the drugability of the CCUG repeat that causes type 2 myotonic dystrophy. My lab's been working on all types of repeat expansion disorders, but the thing that we've really been working on first and foremost are type 1 and type 2 myotonic dystrophy. It's really great always to come to the MDF meeting. This is a special meeting for a wide variety of reasons. It's a first meeting that I went to when I started an academic group, 16 years ago. The thing that is most special about this meeting, and I want all the scientists to recognize that every time they're at it, is having the ability to be together, not only with scientists. I mean, we talk jargon to each other all the time, but really to be together with the patients. And so we missed that during this virtual format. Or in the virtual format. I remember being in elevators with the patients, and that's a spiritual thing that I've taken with me each year. I come to these meetings, and so I miss that. And I hope once we get past this COVID thing, we all can get together again. And I think that's really important. It's certainly, when you're doing basic science research that you hope one day can go to a patient, it's oftentimes you don't really have a sensitivity to the cells that you have come from patients and their families, and they give that to you. And so this is the one meeting and all the meetings that I've been to in my scientific life that is most special, because it's, you breathe the same air, you are in the elevators with these patients. And I hope we can get back to that in 2022. I'm a representative of a group of 25 individuals that are currently in my lab, but about 120 that have been in my lab throughout the years. Here's a collection of them. They range from graduate students and postdocs to staff scientists. I don't want to take credit for this one quite yet, but I've had fun with Yu's daughter, and that's special too, is to be able to be in these people's lives. We've also been blessed to have resources to support these people. Some of those resources have come from the Department of Defense, and the Myotonic Dystrophy Foundation has done a remarkable job in getting government funding by the DoD and the Department of Defense that we've been blessed to have, and a collaborative grant with Eric Wang that you all know. We've also been blessed to have funding by the US taxpayer, by the National Institutes of Health at various flavors. Oftentimes people ask, why do we care about funding basic science? And I realize this talk is to scientists, but think about COVID vaccines. Within a year of us sequencing a genome, we were able to design and deliver a COVID vaccine, which is amazing. And the reason we were able to do that is because the basic science that a lot of us probably take for granted in the room has been able to deliver that. Not only could by Studier and Uhlenbeck synthesize RNA, but we knew genome sequencing, how spike proteins worked, how RNA folds, how to not have RNA be degraded, how to manufacture and deliver them in nanoparticles. I mean, this is just remarkable. And it's remarkable in part because of the people that have been able to do that. So in today's talk, I'm going to describe work that's mainly been done by Raphael Ben-Hamiu, who's currently an assistant professor at the University of Jerusalem. Raphael was supported by an MDF postdoctoral fellowship. I think that's one of the things that we need to recognize, that our work here that we do, especially in academic labs, gets amplified. And it gets amplified by work that people like Raphael will do in his academic lab. And also people like Ali Angibello, who's currently a scientist at Expansion Therapeutics, is currently doing to deliver medicines to patients. There's 3 papers that I'm mainly going to focus on in this talk, 2 sort of discovery papers in ACS chemical biology and cell chemistry and biology that were published in 2020. And then I'm also going to talk about more recent work in the Journal of Medicinal Chemistry that was published a few months ago. My lab has solely worked on drugging RNA with small molecules. I don't have to tell this group why we're interested in it, but RNA plays a really important role in disease processes. About 1% of human proteins are encoded by your human genome, and yet about 80 to 90% of your human genome is encoded into RNA that can have a whole host of activities and function that can be both coding and non-coding. And we think about druggability, about 15% of our proteins have been drugged with a small molecule that binds to a fold in that protein. And basically about 0 of our human RNAs have been drugged with small molecules. That being said, those in the therapeutic development area here are really standing on the shoulders of work of Frank Bennett, Stan Cook at Ionis Therapeutics, and Charles Thornton, and others that have worked in the area to deliver antisense oligonucleotides that target these RNAs. Yeah, it's a really promising approach. There's a wide variety of other people that are working in that area. They're using a lot of the foundations of basic science that have been done by not only Charles, Tom Cooper, Mari Swanson, Laura Random, a collection of individuals to try to deliver medicines to these patients, and Charles Thornton. So I think we all as a community need to recognize that a win that we would have in this scientific enterprise that we're in generally is a win for science. It's not a win for an individual, and it's a win for patients, no matter what the modality is. RNA, we think, is a druggable entity with small molecules because it simply plays a role in, every disease known to man, including things like muscular dystrophy, which is what we've been focused on. The way that my group and I have gone about targeting RNA with small molecules, I'm probably boring many of you to tears that have heard me talk, but the way we look at it is RNA has a sequence, which is shown to the left, and that RNA sequence collapses into a structure that's shown to the right. And this is a general way of looking at RNA, is that RNA sort of adopts two global molecular recognition problems. One is if there's a region of an RNA that's not abundantly structured, then those RNAs are targeted by using base pairing. Watson-Crick or Watson-Crick-Franklin more recently base pairing, where you can recognize A, C, G, and U in an RNA target. You can use a complementary oligonucleotide to bind to that region of an RNA and disable it. What our lab has been doing and has worked on for 12.5 years has been looking at regions of RNA that have a high probability of structure and delivering compounds that can bind to these RNA structures to affect biology in defined ways. Now, there's reasons why you'd want to have a small molecule do this, and those reasons include you can have compounds that wouldn't have to necessarily be injectables, that could be orally delivered and could get into patient affected tissues more broadly. These molecules could be orally delivered and cross the blood-brain barrier. There's a lot of challenges in this, and these molecules could bind to RNA targets and affect their biology in very unique ways that could, not only help us study the biological pathways that the RNAs are involved in, but be able to leverage those pathways with molecules and try to, therapeutics that we take as pills to be able to affect their biology. And so we have a lot of work in this area. Once we identify an RNA target that has a high probability of structure, we could ask fundamental questions and go, is it a druggable pocket? When we identify ligands that bind to that pocket, we can then see how it affects biology. What I'm going to tell you is an interesting story, or at least what I think is an interesting story. I don't want to judge what your perception of this talk is for targeting the DM2 RNA. Okay, and so there are many challenges in targeting RNA with small molecules, despite the immense opportunities. So one challenge is that people go, well, RNA is only built of four bases. Proteins are built of 20 amino acids. You're not going to be able to target it specifically, and RNA is not going to robustly fold. And so I'm going to tell you that for repeat expansions, basically all of them, where it gets made into RNA, they have robust folds, and those robust folds are shown here. These toxic RNAs in DM1 and DM2 form these very robustly folded structures that we call stem loops or hairpins, and those things can be targeted with small molecules. And the second thing to the right is that many people have said RNA cannot be targeted with drug-like small molecules that can be delivered orally. I'm going to sort of challenge that assertion here. Here's one of the molecules that we've delivered that can target DM2 RNA. It looks like a regular drug. And we're going to continue to advance these molecules and others within expansion therapeutics. It really, I'm in an academic institution. You really have to have industrial scientists like Valerie Cullen, Renato Skirl, Andy Good, Mark Ledboer, Suzanne Zuchek, Cy Velagiputi, Ali Angebello, to really industrialize the discovery of these molecules. But my job here is to deliver lead compounds that can probe the biology, and then that goes to an industrialized setting where you really need all the resourcing that expansion has to deliver medicines. And so RNA repeat expansion causes many biological processes, DM1 and DM2. I'm going to talk about DM2 today, is that you get a repeating RNA. That repeating RNA binds to proteins. Proteins, in the case of myotonic dystrophy type 1 and type 2, are muscle-blind like 1 proteins, where you actually get these RNA protein aggregates and you titrate out the protein and you basically disable or you limit its availability to affect normal biological processes. For the case of DM1 and DM2, sequestered muscle-blind causes RNA processing or splicing defects throughout a patient. So these can range to CNS defects, to also issues in the heart, the skeletal muscle, etc. And so again, in DM2, what happens is this long and toxic RNA repeat that's in intron 1 of the CNBP pre-messenger RNA is bound by muscle-blind. And muscle-blind in a normal setting, it'll bind to these RNA structures to control exclusion and inclusion of exons. When you have DM2 or DM1, these RNA structures in the pre-messenger RNAs that are normally coded by muscle-blind are not bound by that protein, and you wind up affecting RNA splicing, as you all likely know. The other thing, and this was discovered in Mari Swanson's lab by Lucas Schneider in a PNAS paper, and I think 2018. Sorry, Mari, I had your reference here, and now it's gone. But nevertheless, it's a very interesting paper where they asked a question, and the pitch there was this could be a biomarker for these patients. Is that when, because you could ask a question and go, well, this DM2 is an intronic RNA. How does an intronic RNA cause disease? You would think it's spliced out and degraded. And so what they found is that these intronic RNAs, when they're GC rich, they're harbored as retained introns. So that means that intron is part of a messenger RNA, and it's protected from being degraded by having all of the other parts of the messenger RNA associated with it, a poly A tail at the end, etc. And so what Mari showed is that when you have DM1, you can get an enhancement in the retained intron, and there's a fraction of this toxic RNA that's protected from being decayed. This is where Raphael came in. So we asked a question and said, well, why are we getting this? Why is Mari and Lucas Schneider, why do they observe that these introns are retained? And so it turns out that we think that this retention is due to this toxic RNA binding muscle-blind. And so when muscle-blind is bound, and this was validated by both a gain of function where we transfect in muscle-blind, or we use an siRNA to knock it down, is that when you knock down muscle-blind, you can eliminate the retained intron. When you add more muscle-blind in, you get more of the retained intron. And so that suggests that binding of proteins to these toxic RNAs triggers intron retention. And so it could be that the spliceosome doesn't recognize this protein-coded repeat expansion as being an intron, and that gets retained, and you get defects in RNA splicing. And so one question we had is, could we affect all of these DM2 associated defects in a very positive way by having small molecules that are drug-like? And so you could imagine you could have a small molecule binding to this toxic repeat expansion. It could kick off muscle-blind. Muscle-blind would then be able to affect normal RNA splicing. Another question could be as if you bind to this toxic RNA that's intronic and you kick off muscle-blind, could the spliceosome now recognize this intronic RNA, liberate it, and it would no longer be a retained intron, and then could it be subjected to normal decay pathways? And in that way, you could have a binding small molecule that would have rule of 5, all these properties that we're interested in, and it could be a degrader. And so what we did is we asked if that could be possible by using drug-like small molecules. And so what we did is we compiled small molecule collection, about a 12,000 compound collection, by analyzing the features in all known small molecule small molecules that bind RNA by a software that we have that's called Inforna. This allowed us to identify what are the features in drug-like small molecule in molecules that bind RNA. We could then compile 12,000 molecules that have these features that are within drug-like space. And so what we did is we compared from drug bank, which is shown here in gray. So this is an analysis of various physical chemical properties, molecular weight, C log P, hydrophobic surface area, rotatable bonds, etc., and compared that to our RNA focused library. And you can see that all of these are similar, is that these molecules and these libraries that we identified have features that are similar to those that are observed in known drugs. And so what we did is we screened these compounds for their ability to inhibit the RNA protein complex formation. We can get various compounds that inhibit with less than 50 micromolar. These are just lead compounds. When we deliver some of these compounds, these are these quinozoline core molecules. They can modestly affect RNA splicing defect and insulin receptor splicing that we observe in this DM2 cell line modestly. So micromolar concentrations of those compounds can affect about 50% of the splicing defect. What we then did is we did medicinal chemistry within these lead compounds, and we can functionalize them at various ways. We can functionalize from this core. We can have R groups here, and we can improve activity from 50 micromolar ability to inhibit this RNA protein complex. We can get it down to 10 micromolar. We can make various derivatives to molecules that have R groups here, and we can get them to low nanomolar activity or 100 nanomolar ability to inhibit this RNA protein complex. So we can do medicinal chemistry where we can functionalize these molecules in various ways to improve the affinity and the ability of these compounds to inhibit the formation of these RNA protein complexes. What we did is by screening these molecules for their ability to inhibit these complexes, but also for their activities in cells. And all this data is published in that JMedChem paper in 2021. We could get clusters of molecules that have various functionalities that didn't have any observed toxicity in these patient-derived cell lines, which we would throw out any molecules that have toxicity. And then we could hone in on molecules like compound 63, whose structure is shown here, which could improve about 50 to 40% of the RNA splicing defects in these DM1 cells at about 1 micromolar compound concentration. And these compounds had no effect on a healthy cell for splicing of for affecting inclusion of exon 11 splicing defect in this IR insulin receptor pre-messenger RNA in the DM2 fibroblasts. When we studied this lead compound 63, we gratifyingly found that it had a dissociation constant for this toxic RNA repeat of about 190 nanomolar. When we used an RNA that was completely base paired, we found that there was greater than 20 micromolar KD for this compound. That is, it's very specific for the toxic RNA repeat over a base paired RNA and even DNA. So you can indeed get drug-like molecules. These molecules are so drug-like, they have properties that suggest they could be orally delivered and cross the blood-brain barrier. So you can get 100 nanomolar dissociation compounds that affect DM2 biology with this modest lead in cells. What they can also do is disrupt in DM2 cell lines the formation of these toxic RNA protein complexes, and it can disrupt them, and you can see that you eliminate them in these DM2 cells. DM2, we need a lot more work on basic biological systems, I think, to really drive medicines for this disease, which can include more robust cell lines. We're working in fibroblasts here, but we'd really like to have myotubes, also iPSCs to differentiate into various cell lines. I know people are working on this, Laura Ranum, Charles Thornton, and others. We'd also like to have a mouse model, which we really don't have now to drive medicine development. And the Charles Thornton, Tom Cooper, Genevieve Gordon mouse models, I think, and others that people like Dine have made are really important for driving medicine development. And I think we need more investment in that for DM2. What's interesting is that we knew that when we designed these and other molecules, they would disrupt muscleblind from binding to the target, and they would also eliminate the retained intron. And so if we use primers, we know that when we take these compounds that bind to this target, they'll eliminate muscleblind from it. That'll liberate that retained intron and it'll be degraded. And this degradation occurs just at that CCUG containing intron. There's no effect on native CMBP or the mature messenger RNA that harbors that intron. And we did a wide variety of studies to show that this degradation occurs because you liberate that retained intron and it gets shunted to the RNA exosome. And it's that nuclear RNA exosome that when you use siRNAs or CRISPR, we've done this for other targets, when we knock that out, we inhibit the ability of us to eliminate the retained intron. We also know if we knock out this spliceosome, we don't get liberation of that retained intron. So it's basically muscleblind protects that intron from being recognized by the spliceosome. We have a small molecule, you disrupt the formation of this RNA protein complex. That causes splicing and debranching to liberate that intron. What I'm showing you here is for Fuchs' corneal dystrophy, but the same mechanism occurs for DM2 in the CMBP. That causes this RNA to be decayed by the exosome. And we've found that this mechanism is general for all toxic repeats that are present in introns, including CUG repeats in Fuchs' corneal dystrophy. It's not intronic in DM1 and for DM2. So a binding small molecule can become a degrader. The reason that we also think this is important is that when you go to use at least CUG targeting ASOs for DM2, is other genes, other messenger RNAs have short stretches of CCUG repeats, such as not only the mutant of CMBP, but also AGO and muscleblind itself has a CCUG repeat. And so we published this in an ACS chemical biology paper. And so if you use ASOs that target these repeats, you can eliminate not only CMBP and the mutant version, but you can also deplete muscleblind. So in that way, when you go to use an ASO where you want to eliminate a toxic RNA, you also functionally deplete muscleblind and can enhance some of the defects. This is at least for a CUG targeting ASO. Other ASOs can be developed to target this effectively. That's what we've observed. And so it suggests that a small molecule targeting strategy might be very advantageous for DM2, even at the functional level, in addition to the PK or distribution reasons that we're talking about. And I have to say that these retained introns, it's very sensitive to eliminate them with these small molecules. So this might even articulate a selectivity strategy for these compounds for targeting at least repeat expansions that are harbored in retained introns, which could be Fuchs' corneal dystrophy, a CUG repeat, DM2, a CCUG repeat, and even things like C9orf-associated ALS. So I want to sort of end here and say a couple things. I think doing science is fun with people with many perspectives. For the past 2 years, we've had to deal with COVID. One of the, like many of you, I have 2 young children at home. So one of the things that kept my family grounded is having the ability to interact with young people, and young people that come from very different perspectives around the world. I didn't think about it when I got into science, but having people from China, from India, even from Canada, it's a joke, and the United States. I'm a kid from Baltimore, so I never thought I would, despite having the last name Disney, live in Florida. But to have people from different states and different countries work in labs is a real blessing. And I recognize that more and more as time goes on. There's also several ways to degrade an RNA by using small molecules. I'm showing you this intron retention RNA exosome method, but we've developed ways in which you can unnaturally affix ribonucleases to RNAs by using small molecules. And structure targeting compounds can target mutant alleles and repeat expansion disorders. And as many of you know, science, one of the great things about science is that the challenges and the discovery process never ends. There's always more to discover. And so affecting RNA biology with structure-specific compounds can not only allow us to understand RNA biology, we didn't understand, this intron retention mechanism or how you could affect it with small molecules. We're standing on the shoulders of Lucas Schneider and Mari Swanson. But we want to be able to tap into this approach to have it positively impact human health. And, these and other approaches need, I think, to be pushed to patients. So Biogen Ionis has ASOs. There's a lot of work in using ASO peptide, ASO antibody conjugates to get more delivered to muscle. We have structure binding compounds with small molecules that can target these RNAs. And I think all these promising approaches, no matter what bias you're coming from, I'm an RNA structure guy who likes small molecules, and others are oligonucleotide people that have antisense that target these toxic repeats. They all need to be pushed into patients to positively affect humans. And so I think, we're very happy to be able to do that. I'm very happy to be able to talk at this session. And so thank you very much. I'm happy to answer any questions that you have. I'm not hard to find, and I probably have an easy email to remember, disney@scripps.edu. So thank you all. I appreciate it very much.